Hi, welcome back to my channel. Me, Sizin, a clinical geneticist, medical doctor, Istanbul, Turkey. Today, I'm thrilled to introduce a comprehensive three-part series summarizing the entire textbook, Human Molecular Genetics by Tom Strachan and Andrew P. Reed. We will be focusing specifically on the fifth edition, which was officially published in 2019. Because this book is so massive and packed with crucial information, I will be presenting the entire book in three main parts to make it easier to digest. However, before we dive into the first part, I want to emphasize a very important point. While these videos will give you a fantastic and logical overview of the concepts, the absolute best way to truly master this subject is to study directly from the textbook itself. No summary can replace the depth, details, and the rich framework of principles provided in the original book. Think of this series as your roadmap, but I highly encourage you to grab a copy and read the book firsthand. Now, if you are ready, let's jump into part 1. Let's start with the absolute basics, DNA and RNA. In all cells, the DNA serves as the hereditary material. The core concept you need to know is the central dogma of molecular biology. Genetic information generally flows in a one-way direction. DNA is transcribed into RNA, and then this RNA translates to synthesized proteins at the ribosomes. This process is what builds and operates every single cell in our bodies. Now, this massive amount of DNA isn't just floating around randomly, it is highly organized into chromosomes. Humans typically have 23 pairs of chromosomes, which consist of double stranded DNA bound to proteins. When our cells divide for growth, they use a process called mitosis. But to produce sperm and egg cells, they use meiosis, which introduced genetic diversity. Thanks to Gregor Mendel's work in 1860s, we understand that many traits and disease follow Mendelian inheritance patterns, meaning they are determined by a single genetic locus. In 2003, science achieved something incredible. The Human Genome Project successfully mapped our genome. We learned that the human nuclear genome consists of about 3.1 billion base pairs. But here is the most shocking part. Despite our complexity, we only have around 20,000 protein coding genes. In fact, protein coding DNA makes up just about 1.1% of our entire genome. The rest includes regulatory elements and non-coding RNAs that sophisticatedly control how and when our genes are expressed. That's it for part 1. Today we covered the obsolete basics of DNA, cells, and the human genome. Next time in part 2, we will talk about what makes us all unique, genetic variation and human evolution. See you in the next one. Thank you. Bye.